Okay, so don't worry, you're at the right talk. I just wanted to start motivating my lectures today by showing you these equations. So this is the Lagrangian of the Standard Model. These are the general relativity equations, which may be really an epiphenomenon of this in some way. And at some point in time, yes, I worried about these equations, and I thought I'd start by telling you why I don't worry about them anymore, why I decided to work in another field. And so basically, from the point of view of a theoretical physicist, science is sort of almost done. There are some gaps and holes to fill and some inconsistencies to resolve, but basically, we think that these equations cover about almost any observable phenomenon around us. And so in principle, if you were to you start out with a hot, small, concentrated system right after the Big Bang, and you run these equations forward in time, you'll get a universe, okay? That's a recipe for you, for running in a universe. So in principle, I think we all agree, or we should all agree, that's what we believe. But if I look at these equations, there seems to be something sort of missing to me. So to me, at least, it's totally not obvious that if you take these equations and run them forward in time, that you're going to get things like this, eventually, that's implied, or this, or even that. Okay? I think, to me, it's a very big mystery why such equations of motion apparently seem to apply that one gets these things over time. And so this is really what motivates, on a sort of very fundamental level, the questions I work on. I want to understand, how can I understand the emergence of complex systems with complex behaviors from the underlying physical laws. And in particular, I was attracted to the field of gene regulation and gene regulatory networks. And the reason why I found this an attractive field to work in is because gene regulation underlies much of the complex adaptive behaviors that biological systems exhibit that dead systems, non-living matter, do not exhibit. So one example is, for example, it allows cells to respond on their own behalf to changing environments. And when I say on their own behalf, I mean is that if you have here this bacterial cell sitting in the blue environment, and the environment changes to the orange environment, it may sense this, change its internal state from this white state to the green state, and this green state now allows this cell to replicate in this new environment, whereas it could not replicate in this white state. Right? So in this sense, the change that this cell undergoes is a change on its own behalf. Sorry? No. I don't have a phone. Sorry about that. Another thing that gene regulatory networks allow cells to do is it allows multicellular organisms to express a single genotype into a very large array of very diverse phenotypes. So these are all pictures of human cell types. And it allows these multicellular sort of societies of cells to form. And so that's one thing, it underlies many of the complex behavior. And the other thing is that the basic physical mechanisms are clear. Right? So there's no mystery about what are the physical mechanisms underlying gene regulation. And at the end of the day, very, very roughly speaking, it all boils down to the recognition of short DNA sequence fragments by proteins. And the attraction of this is that this is directly encoded in the DNA. And so therefore, mutations on the DNA level immediately affect the binding affinity of these proteins to their binding site. And so this gives me the idea that there might be some hope to even understand how such regulatory systems evolve. Whereas if we're talking about, let's say, a complex enzymatic network, where mutations in a protein first is going to change the folding of the protein and its precise state, which is going to change kinetic parameters and so on. The step from genotype to phenotype is much, much further. All right. So let's just recap very quickly with this cartoon how transcription regulation works. And this picture here mainly applies to bacteria, but the cartoon sort of captures transcription regulation in general. So there are transcription factors, proteins in the cell that bind DNA in a sequence-specific manner. So some sequences are more strongly bound than others. And these transcription factors often have such binding sites near starts of genes. So here in this cartoon, there is a gene here. Here, this little arrow is where the transcription starts of this gene. And in the absence of any transcription factors, the RNA polymerase has just some basal rate at which it will bind to a binding site immediately upstream of the transcription start and transcribe this gene. And so this gene will be transcribed at some rate. Now, in another condition, there may be another transcription factor expressed here, this green one, that has a binding site just upstream. And when it binds to this site, because this transcription factor has an interface that is sticky to this polymerase, it actually increases the rate at which the polymerase now binds to this site and transcribes. So you upregulate transcription. There may be another transcription factor that is expressed in another condition, this red one here, which binds to a site here. And when this red transcription factor is expressed and binds, the RNA polymerase can no longer access this site. And now the transcription is repressed simply by getting in the way of the polymerase binding to its site. So this is sort of a very cartoonish way of how you can imagine the presence or absence of these transcription factors changes the rate of transcription of a gene. All right, so to show you sort of how this works in action, I'll just show you what might be considered the hydrogen atom of gene regulation. So this was the system in which gene regulation was originally discovered by Jacob Monod. So there's a paper from 1961. It's the lac operon in E. coli. So E. coli is a bacterium, lives in your gut. It has a set of 3 genes that code for proteins that are used to metabolize lactose. All right? And it additionally has a transcription factor called LAC-I that represses transcription of the lac operon. So this repressor binds here and stops the RNA polymerase from transcribing these genes. Now, this is the condition when lactose is absent. When lactose is present, this small molecule can actually bind to this repressor. This will change the conformation of this protein, the binding. In this new conformation, this repressor can no longer bind to its site, so it will let go of the DNA. This now allows the polymerase to go and transcribe the genes, and so you will now make the proteins for eating lactose. As long as lactose remains present at high enough concentration, this process goes on, but at some point, if the lactose runs out, of course, this repressor will no longer be bound by lactose, will come off, the repressor will go back into a conformation where it binds DNA, and it will shut the genes back off. So this is this very nice system that allows the cells to respond to the presence of lactose, make the genes that it needs for eating the lactose, and when the lactose goes away, turn these genes back off. All right, so that's the very sort of fundamental idea of how gene regulation works. And how this is encoded on the DNA, in the case of the lac operon, so there are these 3 genes, LAC-Z, LAC-Y, and LAC-A, that are involved in metabolizing lactose, and there are 3 sites on the DNA. One is for binding of the polymerase. Sorry, no, the red one here is for binding, what am I saying? Yes, the red one is for binding of the polymerase. Upstream of it is a binding site for an activator protein called CRP or CAP. This protein is expressed when the glucose levels are low. So E. coli likes eating, well, it likes drinking Coca-Cola better than it likes drinking milk. So it prefers glucose over lactose. And so only when glucose levels are low, this activator will come and boost the expression of these genes. And the second site here is the binding site for the lac repressor, and this makes sure that this operon is only expressed when lactose is present. So the summary that I want you to get from this short cartoonish introduction is that, roughly speaking, the constellation of binding sites in the promoter of the gene determines how the gene expression is regulated as a function of the concentration of transcription factors that are present in the cell. Right? So the idea is, in the cell there is a certain combination of concentrations of transcription factors. There is a certain constellation of binding sites in a promoter, and this determines now the output from the promoter. Yes, Sanjay.
So in this picture, it seems, but that might not be correct. Just want the clarification that the LAC-I repressor is binding at the same place where the RNA polymerase was supposed to bind. That red.
Yeah. Okay.
That red rectangle.
So I think what these people that made this picture tried to do, I didn't make this picture, I stole it from somewhere, is that the polymerase has a foot, which is called a sigma factor. It's a part of the complex that is the polymerase in bacteria. And that binds to this black site here. And the binding site of this LAC-I is just a little bit downstream, and actually is going to be the location where most of the RNA polymerase molecule will be located when it's initiating to transcribe the DNA. So this is why this configuration looks like that. And it captures the idea is that when the LAC-I repressor is bound, it really gets in the way of the polymerase. I don't know, operator site. People use different names, but you should think of these as just short fragments of DNA that are recognized by these proteins. Yes.
Roughly, what is the difference between the bacterial transcription and eukaryotic transcription?
Oh.
I have no idea, so.
Well, I'll get to some of this tomorrow, hopefully. But so one fundamental difference between eukaryotes and prokaryotes is that in eukaryotic cells, the DNA is wrapped in these sort of histone octamers called nucleosomes, and there's this chromatin structure which doesn't exist in bacteria. In bacteria, it's also the case that all gene regulation, all binding sites that are relevant for the expression of a gene occur near the start site of the gene, so just within a few hundred base pairs of it. Whereas in eukaryotes, as John in his lectures pointed out, there are mechanisms by which binding sites that are far away from the promoter can regulate the expression of the gene. But additionally, in E. coli, you really just need a polymerase and DNA and the right material to get transcription going, whereas in eukaryotes, there are often lots of cofactors involved and mediator complexes, and it's really there's a zoo of molecules that are involved in transcription initiation in eukaryotes. So it's more complicated in general. Yeah.
The E. coli polymerase essentially has two polypeptide components. The eukaryote polymerase has essentially over 100. It's kind of insane.
Yeah, sorry, a quick one. Where is the role of enhancers here and silencers? So where do enhancers and silencers come into play here?
So they come only in eukaryotes.
Right.
All right. So with this was my introduction of the topic for today. So this is an outline of the lectures that I'm planning. So today I would like to give a fairly technical talk about computational methods for predicting constellations of regulatory sites in DNA. Right? So with the introduction, I hope to have convinced you that a good first step towards understanding these gene regulatory networks is just simply map where are all the binding sites in the DNA for all the transcription factors. Right? When I moved into this field, I thought, I just have to work out a good method so that I can map all the binding sites for all the transcription factors genome-wide, and then half of the problem has been solved, and we can move on from there. So this is what I'll be focused on today. If I have time, I will also tell you about some ideas of how you can go from such constellations of regulatory sites to genome-wide expression patterns. Tomorrow, I will talk to you more about evolution and large-scale patterns in genomes and also in gene regulatory networks, and what we can learn about sort of large-scale organizing principles of such networks. And I will tell you about gene expression noise and the role of gene expression noise in the evolution of de novo gene regulation. And then if I have time left, if I don't need the last day to cover this topic, I will tell you about some recent work about how bacteria evolve in the wild and what actually is the meaning of a phylogenetic tree. All right, so that's the plan. All right. So let's start about mapping transcription factor binding sites in genomes. So the question you first have to ask, what do you actually mean when you say that some here in the DNA, right? So in this picture, we put these little tablets here that said there is a binding site for this protein and there is a binding site for that protein. What does that actually mean? So what's the physics of this process? What does it mean to say there's a site here? Well, at the end of the day, the interaction between a transcription factor and its binding site is essentially characterized by two parameters: the strength of the binding energy of the interaction between the transcription factor and the binding site, and the concentration of the transcription factor. So in the simplest statistical physics sort of thermodynamic model, you could calculate what's the probability, what's the proportion of time that a particular site is bound by a transcription factor as a function of the concentration C of the transcription factor and the binding energy E, so how much is the change in the energy of the state when the transcription factor binds to the site, and it's given by such a Fermi function. All right, so as you're changing the log concentration of the transcription factor, you go from being almost certainly unbound to being almost certainly bound. All right, so that's clearly a continuous process, right? As you continuously change the concentration of the transcription factors, the fraction of the time that transcription factors will be bound at different sites is going up and down according to such functions. So in principle, it seems a little bit strange to have this sort of binary partition of the DNA into places that are not sites and places that are sites, right? Because in particular, if you make the concentration of the transcription factor high enough, eventually it will coat the entire DNA. All right, so what are we thinking if we say that we can call things sites or non-site? Well, what we're sort of thinking is that the transcription factor itself doesn't scan all possible concentrations, but that in the normal operating of a cell, a transcription factor is either expressed in some low concentration range, maybe sort of this range, or in other conditions, it's expressed roughly in this range. So that there are particular ranges in which this transcription factor is expressed when it's off and when it's on. And so now you can basically define binding sites as all the places in the DNA that have binding energies such that they are bound almost all of the time when the transcription factor is expressed, and they're not bound when the transcription factor is low expressed. All right, so that's sort of the rough way in which people like to think of classifying. Yes?
I'm going to apologize for my lack of statistical mechanics, but is this equation coming from assuming that DNA is in one place and transcription factors are shorter than a distance? You know, I mean, they're, I guess, everywhere on the cell where, I mean, and they don't move, they just randomly move, right? Where is this probability coming from? How, I mean...
Yeah, so there is different ways that you can derive such equations. I mean, I can't give a whole sort of course on this now, but basically you can either think about it dynamically, and say the concentration is just the density of the transcription factor in the cell, and as these transcription factors are randomly moving around, the probability that the transcription factor will meet the binding site is proportional to C. So the rate at which you go from an unbound state to a bound state is proportional to the concentration of the transcription factor. And now the rate at which you go from the bound state to the unbound state is exactly exponential in the binding energy of the transcription factor. So John also talked about this. This is this Arrhenius law for the activate, to get out of this state. Alternatively, you can say, no, I'm just going to do the thermodynamics. I'm just going to look at all possible ways of arranging my transcription factors in the cell and putting them on or off the DNA, calculate the total energy of each of these possible microscopic states, and then say that I'm going to use maximum entropy to say there's going to be a Boltzmann distribution over these microscopic states, and you will get the same answer. All right, so these are sort of the ways you can approach this. Yeah.
Just asking, can you give some interpretation of the normalization factor K? Is it a constant or function of the variables?
Yes.
That function of the variables K, that normalization factor K.
So it's a constant. It's just meant to be a constant.
Just a constant.
Yes.
Okay. So it does not vary with C and D.
No. C and E. So the two variables that I want you to focus on here are C and E. I'm also not going to change the temperature typically, but I want to look at how does the binding probability change as a function of C and E. Yeah. Yeah. Okay, so I know this is one of these things, right? It's like asking somebody to use another toothbrush. So you're used to putting a minus there, but I think it's actually, it's more intuitive to say there is a certain strength of binding, and the binding actually goes up with E. Okay, so when I introduce this to my biologists and so on, I write it like that because I think it's more intuitive. No, that's exactly it, right? So at every place of the DNA, and this is my next slide, so maybe I should go to my next slide. At every place of the DNA, for every sequence S, there's going to be some binding energy, all right? So the question is, what is that binding energy? What kind of function is that? So it turns out that to a good approximation, for the large majority of transcription factors, this binding energy can be written as a sum of contributions from every individual nucleotide in the DNA that the transcription factor touches. So this transcription factor here, let's assume, touches a segment of length L in the DNA, and its binding energies to some sequence S of length L can be written as the sum of binding energy contributions from each position I from the letter SI that occurs at this position. All right? So to characterize the binding specificity of this transcription factor, I just need to give you one matrix of numbers. Namely, I need to give you a matrix of number of columns is L, number of positions that the transcription factor touches, and the number of rows is 4, 1 for each of the 4 letters of the DNA. And once I've given you those L times 4 energies, you can calculate the energy for any particular sequence segments S. All right? And so then, as a function of that energy ES, as I just said, the probability, the fraction of the time the transcription factor is bound, is such a Fermi function. Now, this ignores all kinds of things. It ignores that transcription factors might bind to overlapping places of the DNA and get in each other's way and so on. And one can treat all those things, but to a first approximation, you can think of such functions as determining how often a site will be bound. All right. Now, the key next step is to make the following assumption and to say, if I want to work out what is the probability that the binding site for a particular transcription factor will have sequence S, I'm going to assume that the only thing that constrains those sequences is that they need to have some characteristic average energy E star. Okay, so I'm going to say what makes a sequence a binding site is that they have on average, a certain binding energy, which is typically much higher than random DNA would have. Okay? That's what characterizes binding sites. Now what you can do is you can say, what will be the distribution of sequences that maximizes entropy under the constraint that the average binding energy of these sequences is E star? And that's given by, again, such a Boltzmann form, but here now this Lagrange multiplier in the exponent is not inverse temperature like it is in the Boltzmann equation. Here is just some general constant that you tune so as to make sure that the average of this distribution of sequences, the energies of the distribution of sequences, is this E star. All right? So it basically just says the probability of seeing sequence L is exponential in the energy of the sequence. All right, so I repeated that again here. So the nice thing of that form is that it actually factorizes into a product over the individual positions from I as 1 to L of the probability to see letter Si at position I. So what we can do is, instead of giving you this matrix of energies, instead of telling you for each position I and each possible DNA nucleotide alpha what is the energy contribution, I can actually just give you a matrix of probabilities. I can tell you for each position I, what's the probability that in a binding site, the letter at position I will be Si. All right, so it's just a matrix of, again, L by 4 probabilities. So now, given such a weight matrix, the probability that a binding site for this transcription factor will have sequence S is simply the product over all positions, I is 1 to L, and then the weight matrix entry for position I, letter Si. All right, so it's just this simple product. Now note, this is not the probability that a segment with sequence S is a binding site. All right, this is the probability that if you have a binding site, that it will have sequence S. All right, to get the probability that a sequence S will be a binding site, we need something more, and I will get to that in a second. All right, so just to give you an idea of how this works, here's an example. So this alignment of sequences here is the alignment of known binding sites for the transcription factor through R. It's really like this lac one, but this one is the repressor that turns off the genes for fructose eating. Okay? That's why it's called through R. And here's its alignment. And basically, you construct a weight matrix from this alignment that gives the probabilities for each position to I to have letter alpha. So for example, here in this third column, you might have that the weight matrix entries are that W3A is 27%, W3C is 20%, double G is the most common one, is almost 50%, and T is very unlikely. And typically what people do is they make these sort of graphical representations of such weight matrices where these are the positions of the binding site, and then at each position you see 4 letters, and the relative size of the letter is given by the relative probability of the letters at this position, and the total height of the column is really 2 bits minus the entropy of the probability of letters in that column. That is to say, if the letters in a column are very random, then the height of this column is very low, and if they're dominated by just a single letter, then you make a large column. So it'll show you which positions are important and which positions are less important. All right? And so in terms of such a weight matrix, the probability that you will find sequence S is, again, this simple product. All right, so that's the main introduction. Yes.
Explain this again.
Yeah. All you need to know is the following, Sanjay. There is one matrix of probabilities. Wi alpha, where I is the position that goes along the length of the site, and alpha is the 4 letters. Yes. Because the transcription factor, when it binds the DNA, touches L consecutive letters on the DNA. Yes, exactly. Yes. There are ones that can be flexible and change a bit, but it's actually rare. Okay. Yes. Yes. No. What I'm saying is, if you look at all the bindings, all the functional binding sites for this transcription factor, yes, that occur, let's say, in the E. coli genome. These are all the occurrences in the E. coli genome of binding sites for this transcription factor.
These are known binding sites.
Exactly. Right? No, these are all the known alignment of known binding sites for the E. coli transcription factor through R. And from this, and I will not tell you the details of this, we basically reconstruct this weight matrix of probabilities for different letters to occur at different positions, which are easy to estimate from this kind of an alignment. Yes. Actually, it's more complicated like that than that, but I will sort of skip the details, but roughly speaking, yes. There's some little extra, but yes, you can think of it like this. Yes. Right. So here you see everybody has a T, right? Yes, exactly. That's the only thing I want you to get. For each transcription factor, there is such a matrix that gives you a map that tells you what kind of sequences are binding sites for this transcription factor is likely to have. All right? Okay. So I will now basically go through some methods of how you use these weight matrices to find binding sites in the DNA. And what we're going to use for this is a general Bayesian inference approach. And I just want to make a plug, because since this is a course, for what I consider to be the most useful book that I ever read. So this is the book called Probability Theory, The Logic of Science, by this guy over here, E.T. James, was a physicist. And most of you will have been taught probability theory and statistics as having to do with random events and random variables and things like that. But in this book, the approach is taking that what you should really think of as probability theory is something much more general. And what it is it's the mathematically uniquely consistent extension of logic. So logic is a calculus for statements that are true or false and tells you how to combine them to make other statements that are true or false. And now if you ask, can I extend this calculus so that the entities it works on are not just true-false statements, but are statements that have different degrees of plausibility, where 0 means false, 1 means true, and numbers between 0 and 1 mean different degrees of plausibility. All right? If you ask how to make a calculus that acts on such numbers, such plausibilities, and that reduces to logic, it turns out there's really only one way of doing it. And the basically rules that you get are the rules of Bayesian probability theory. All right? And when you take that view of probability theory, it opens up a much wider array of applications than you would get with the sort of thinking of it as random events and so on. And so I'd like to make a plug for this. I've learned an enormous amount from reading this book and learned about how to apply this kind of methodology in many different situations. But so the general way in which you proceed in the cases that we're going to look at is you have some data set D, for example, some set of sequences, and you first want to generate some space of all hypotheses H that you think could have accounted for your data. Okay, so you have to put yourself in a box. You have to say, this is the set of hypotheses I'm going to consider. then you have to assign prior probabilities to each of these hypotheses. Somehow you have to represent your state of information about which hypotheses are more or less likely. You have to define a likelihood model that says, for each hypothesis, how likely is this hypothesis to produce the data that I saw? And then you can use Bayes' theorem to calculate a posterior probability of how likely is now each hypothesis given my data, and it's just the prior probability that you gave to that hypothesis times how likely this hypothesis is to produce the data that you saw, divided by the sum over all possible hypotheses, their prior probability, times their probability of generating the data. So basically what to calculate the posterior for a hypothesis H, this hypothesis is competing with all the other hypotheses for explaining the data. All right? That is sort of the way in which it works. So let's apply this to the simplest example for binding site prediction. So I'm given a sequence S of length L, and it's known to contain a single site for the transcription factor represented by the weight matrix W. So I assume I know this weight matrix W. I know the sequence specificity, I'm given these probabilities. Now I'm given a large sequence of length L, and I know somewhere inside this large sequence is one binding site. Okay? It's rare that you have all that information, but let's just look at the case where we have this information. So now my set of hypotheses are simply all the positions at which I can plot down this site, right? So if there is exactly one site in this sequence, I have to put it in one of these positions. So that's the set of my hypotheses. And I'm going to assume a priori they're all equally likely. I'm just going to say I have no prior information to like one place better than another. So a priori they all have the same probability. Okay, now what's the likelihood? I have to calculate the probability of my entire data, which is your sequence of length L, under the hypothesis that the binding site occurs at one particular position. So let's assume it occurs at this position from I plus 1 to I plus L. What is this likelihood? Well, the likelihood will be a combination of the probability of this sequence, assuming it belongs to this weight matrix. This we've already seen, right? That's just the product over all positions in the site, and then the weight matrix entry for the letter that occurs at position I plus K. Okay, so that's the probability for this. But now I also need the probability for all these letters, assuming they're not a binding site. So we need another model, which is called a background model, and in most cases, people simply say, there are 4 probabilities, which just gives you the frequencies of letters A, C, G, and T in the whole genome. So for each of these positions in the background, I just put a probability B, S, K for the letter at position K. So the total likelihood is just the product of these 3 terms, these background letters, this site, and these background letters. And the posterior that the site occurs at position I is now this likelihood divided by the sum of the likelihoods for all possible positions. All right? Because the prior, since it was a constant, has canceled out between the numerator and the denominator of this expression. So this often happens if you start from a state of complete ignorance and all your hypotheses are equally likely, they cancel between numerator and denominator, and your posteriors only depend on the likelihoods. So it's really the probability that the site is at position I is the likelihood of the data under the assumption it is at I, divided by the sum of the likelihood under the assumption that it's at any of the other positions. Any of the positions, sorry. Yeah. Yes, so those are just the probability of those letters under a background model. And the background model is just going to be a very simple model that every letter that occurs there, for every so at every position K, there is a letter SK, and there's just the probability BSK for this. So I just have 4 numbers, which are just the probabilities of A, C, G, and T in the background, and I just take the probabilities for all these letters in the background. Why am I doing it? Because probability theory tells me I have to calculate the likelihood of my entire data. I thank you actually for this question, because many times people do probability calculations and they try to get away with calculating only the probability of part of their data under their model and leaving out all kinds of other things in their data. And then they sometimes find things go horribly wrong. And once they go back and very carefully look, they see that if they had from the start actually calculate the likelihood for the entire data set, the mistake would not have occurred. So in this particular situation, I might have gotten away with not putting it. But in general, you really have to calculate the probability of your entire data under your hypothesis, which is not just assuming that this sequence came from the weight matrix, but also saying all this stuff came from background. You have to include it. Okay, now, so this was the very, very trivial situation. Now let's take the first next step, less trivial, and say, I don't know how many sites there are in this sequence. I just have an idea of the density of sites. Okay, so I have some prior information that tells me I expect a constant probability pi per position that the site will occur there. So pi is sort of the density with which I expect sites to occur. Okay, now my set of hypotheses has become much, much bigger, right? Because I can put any number of binding sites. So a hypothesis here is now a configuration of a certain number of positions at which sites occur, okay? So I have to first give a prior over this space of hypotheses. In the prior, I'm just going to put is I'm going to pi to the number of places where a site occurs, and 1 minus pi to the number of places where no site occurs. All right, so capital L minus little l times n is the number of places that are black here. Ni is the number of sites. And so the prior probability that I assign to one of these configurations is just given by this expression. And it's a function of what is the expected density of sites that I put in. And now the likelihood for a configuration is, again, quite simply, you go over all letters that are black in these configurations, and for each of these letters, you assign the background probability of that letter. And then you go over all sites, so in this case, over 3 sites, and for each of them, you calculate the probability of this sequence under the weight matrix. Okay, this is, again, simply given. Yes? No. So all I'm saying is I have this long sequence, and I'm just showing you one possible site configuration. And one possible configuration is to assume that this is a binding site, and this is a binding site, and this is also a binding site. And then for each of these sequences, I can calculate how likely that sequence is under this weight matrix. And so I can take the product of the 3. Okay? So now the posterior for this particular configuration is, again, its prior times the likelihood, which is given by this expression, divided by the sum over all possible configurations of the same expression. Now, here we reach the first point where we seem to have a computational problem, because if I have a long sequence and I allow any number of binding sites to occur, this is combinatorially many configurations that I have to consider. Luckily enough, already a long time ago, people have worked out clever ways of calculating such sums. So I'll just show you how you do this. All right, so I want to calculate the probability of my data, which is really the sum over all possible configurations, the prior of the configuration times its likelihood. So what I'm going to do is I'm going to look at the first n letters of my sequence, and I'm going to calculate the sum of the probabilities of all the configurations just over these first n letters. And now it turns out that you can write down a simple recursion relation for this sum. In particular, any configuration of the first n letter has to end either in the last letter not being covered, right, being a background letter, or the last letter being the end of a binding site. Okay, so if I want to have a configuration for these first n letters, those are the only 2 ways in which it can end. So what I can do is I can say, aha, now the sum of the probabilities of all configurations for the first n letters is the sum of the probabilities of the configurations for the first n-1 letters times the prior probability that the last letter will be background times the probability of this letter under the background, or, so plus, the sum of all configurations up to position n minus L, where n is the binding site length, times the probability to put the binding site down, which is pi, times the probability of this segment under the weight matrix. All right? So this gives me a simple recursion relation, and I can, in basically linear time, calculate this partition sum going forward. I can also do it backwards, right? I can, in a similar way, say, look at the last n positions in the letter and go from the other end. Okay? So you can go both ways. And this way, these recursion relations, so in, let's say in the machine learning community, these are called forward-backward equations. In computer science, it's also called dynamic programming. And it's very closely related to, in statistical physics, what I call transfer matrix techniques, right? But it's all this way of just doing the sum using a recursion relation. All right. So, yes. So now what I've shown you is that once I've calculated this partition sum, I can now calculate the posterior probability for any configuration. So for any possible configurations of sites that I give you, I can give you the posterior probability given this sequence. However, those posteriors are typically not very useful because they're so combinatorially many configurations that any one configuration never has a very high probability. Okay, so you want more sort of summary statistics. For example, you'd like to know the probability that a site occurs at a particular position n. All right, so what is the probability that independent of where other sites may occur, at position n, there is a site? Well, that's simply the probability of the set of configurations, I, that all have a site at position n. And it turns out this is also easy to calculate because any of such configuration can be, the probability of these configurations is simply the sum of the probabilities of all possible configurations for the first n letters, then the prior probability of putting a site down, then the probability of this sequence starting at n under the weight matrix, and then times this, again, the sum over all configurations for the last letters going from n plus L plus 1 to the end of the sequence. But right, in these previous equations, I've already pre-calculated all these partition sums, these fn's and these rn's, so I already have them. And so now I just have to reuse them, and reusing them, I can calculate these probabilities that independent of what happens here or here, there is a site at position n. Okay, so this can also be done in linear time. All right, so now I can also, for example, calculate what is the expected total number of sites that are in this sequence. And I'm just summing those probabilities over all possible positions, and that's just given by this sum. Okay, so I can easily calculate this too. So in summary, if I've given as input a large sequence S, or even a set of sequences S, and a weight matrix, and the a priori expected density of sites, I can calculate in linear time the probability for a site to occur at any position, the probability of any particular configuration of sites, the probabilities of subsequences summed over all possible configurations, and also the expected total number of sites in the sequence. All right, now, yes? How far?
Scope where you need to take care of the combinatorics of so this bar that is there having a specific ordered sequence.
Yeah.
Does that ordering matter, or the degeneracy of, like, if you permute and permute the letters, so you have various.
No, that matters, right? So this weight matrix, I don't know where I have yeah, so this weight matrix is a matrix, so for each position I, there is a different set of probabilities for the 4 letters, right? So the probability of an A in position 3 and position 8 is not the same. So if I permute my letters, I'm going to get a very different...
Thanks.
All right, there was more, Vijay? Thank you, that's my next slide. There was another question, yes.
Quick question. Regarding the background,
Yeah.
You had mentioned there that you have a BSJ. So that sounds like you're trying to you're trying to characterize the probability of a given nucleotide being at any point in the entire genome, right?
Yeah.
So are you, how does one actually sort of come up with that? Because you have to sort of, are you going to assume that it's the same across different sites, or are you going to?
So you're asking me about the background model.
The background model, yeah.
And so I didn't want to spend a lot of time on it, but okay, so. So typically the simplest thing you can do, and that's what I've done here, is, well, actually the simplest thing that you can do is to say the probability for any background letter anywhere is just a quarter.
Exactly. Okay,
that's the simplest thing you can do. If you do that, there is a nice thing, one nice thing about that, is that there is a nice mapping from all calculations I've done now to actually thermodynamic solutions of the binding and unbinding of the transcription factors, right? Because there's no energy associated with an unbound piece of DNA, only with the binding, okay? However, we also know that DNA doesn't look like every letter occurs a quarter of the time, right? So typically A's and T's may occur at a very different frequencies than C's and G's. So what people do is they build background models and they simply go genome-wide and look at the frequencies of A's, C's, G's, and T's and say, those are the probabilities in my background. People also sometimes like to build higher order models and say, what's the probability of an A given that the previous letter was a C? And then they have 16 numbers that they have to estimate. Okay. And you can put this in and you can go even higher models and order models and so on.
So
play games with this, but I think most of the time, it does very little. So I think if you have a model that just have single letter frequencies, for most situations I know about, that survives.
Doesn't make too much of a difference.
Yeah. I'm sure there could be people now that will disagree with me. I will go to the mat saying it doesn't really matter much.
Okay. Thank you.
Okay.
So could you please repeat your explanation of the prior of the pi, just maybe to even that one next.
Yeah, this one, huh?
So what's that again, please?
So I have to now. I'm just not going to say that the site. There's only one site, and then it was sort of like that. It's uniform where it is was sort of obvious. Now I have to say any configuration of sites. So I can put either no sites at all, or one, or I can put 2, or 3, or 4, or any number I can fit. And I have to give some prior probabilities to those configurations. And I'm going to basically parameterize my prior information by saying I have some expected density of sites. So I expect some density of sites. On average, in my large collection of data, I expect one site with frequency pi. If you assume that, then this is the prior probability for such configurations. It's just pi to the number of sites, 1 minus pi to the number of black letters. Yes, exactly. Yeah, so you can build, if you have prior information that say sites like to come in clusters, you could build a model that takes such information into account and so on. And there are versions of these methods that do such things. Yeah.
Is there any issue with considering perhaps binding sites that overlap with each other? Like here you're considering the fact that they have to be distinct.
I've taken care of the overlaps. Because when I did this partition sum, notice that to have a configuration that ends at position n, I'm not allowed to put a thing that is sort of halfway overlapping this.
Okay.
All right? And so in a sort of under the hood, right, you can't really see it very explicitly, but this exclusion that sites cannot sit on top of each other was already taken
But
account.
how about in the biological context? Is that seen as in that there are no like overlapping binding sites, or is that?
Well, okay, so there's just the issue of steric hindrance, right? So when one transcription factor is occupying this spot and binds in this groove of the DNA, then typically another protein cannot sit there also.
Okay.
But of course there are cases where one transcription factor can sit on one side and the other one can maybe sit on the groove on the other side, or maybe they have some way to make room for each other. So like in biology, nothing is perfectly true. But most of the time when people apply such models, the assumption is made that only one transcription factor can occupy a particular segment of DNA at one time. Okay. All right. Okay. What if I do not know pi? What if I have no idea what the density of sites in my sequences is? All right. So formally, what probability theory tells me to do is to say, well, you have to come up with a prior that represents your a prior information of what the density might be. And maybe I'll say, I have no idea what the density might be. It's uniform on 0, 1. Or maybe I have some idea that it's most likely in some range somewhere between 5% and 20% or something like this. And I could have a prior that. And then what I have to do is I have to multiply that prior by times the probability of a configuration given the prior and the data. And then I have to integrate over this unknown prior. All right, so in the general rules of probability theory, if your predictions depend on a parameter that you don't know, what you do is you give a prior of this parameter, and then you integrate over it. Okay, so you integrate it out of your probability. Okay, so that's what we would like to do here too. Unfortunately, these integrals over this prior with this, I do not know how to do. I don't think anybody knows how to do those integrals. Okay, so there was this clever trick, right, with this partition sum of doing these many combinatorially many states in a recursive way. But once you've applied this trick, the pi is now sort of convoluted into these recursion relations, and there's no way anymore to simply do this integral. However, in many cases, this integral over pi will be sharply peaked at some pi. So your data basically says the data is much better explained with one particular value of pi than almost any other value of pi. So when that's the case, then this integral can be well approximated by simply finding at what pi the probability of the data is maximized. All right, so you simply ask what pi will maximize the total probability of my data. Okay, so here it is. I'm going to take the logarithm of the probability of my data. So this is the sum. I'm going to take the derivative with respect to pi. Okay, so I'm going to get a 1 over the probability, right, because there's log. And now I write out this probability as the sum over all configurations, the probability of the data given the configuration, times the prior, but only the prior depends on pi. Okay, so when I take the derivative, this derivative only goes here on this term because this doesn't depend on the prior. Now, what was this prior? The prior was pi to the power number of sites in this configuration, 1 minus pi to the number of non-sites, right? The number of black letters, background letters in this configuration. So I have to take the derivative with this. The derivative of this with respect to pi is just the original function, but multiplied by n and divided by pi, right? So if I take the derivative of this, some n comes down and I divide by pi. And when I take the derivative of that, an L minus n little l comes down, and a 1 minus, and I divide by 1 minus pi, and I get a minus sign because of the minus. All right, so now I have to do this sum. But notice, this prior times the likelihood divided by the total probability. That's just the posterior of this configuration. So I'm now just summing over all configurations, their posterior probability, times the number of positions inside, that number of sites, minus positions that are not side. But this sum is just the expected number of sites in the sequence. And this is just the expected number of background letters in the sequence. So basically what I find is that at the optimum, pi must obey that the expected number of sites divided by the expected number of sites plus the expected number of background letters equals pi. So it's almost like a consistency equation. When I'm at the best pi, the actual expected density of the sites matches my prior. Okay? This is what you often find in these kind of models. And so you can find this optimum using a simple procedure which is known as expectation maximization. So you start with some guess, you calculate the expected number of sites and the expected number of background letters, and I use those to set a new value of pi, and I keep going around this loop until pi no longer changes. And it turns out it's guaranteed, it's convex, it will have a single optimum, and so on. So you can find the optimum of pi in this way. Yes?
In that case, how do the different level energy associated to the different binary site contribute to the transcription rate?
That's something that I, looking at the time, will probably not get to. But so typically we don't know. All I know at the moment is now different sequences, how strong do they bind to a transcription factor and how often are they going to be bound. How much that is going to change the expression of the gene, I have not said anything about that yet. And most of the time people don't really know. So that, yeah, okay. Okay, so a last thing that I just want to notice is that all this generalizes trivially to an arbitrary number of such weight matrices. Okay, so if I have a whole collection of weight matrices representing the binding specificities of a whole set of transcription factors, I can also do these partition sums, right? Now the probability of a sequence up to position n is just the sum over all possibilities with which I can end the sequence. So I can either end with a background letter, and you can maybe think of a background model as just a weight matrix of length 1. It's only extension 1, right? It either ends with a background letter, or with a site for the red transcription factor, or with the site for the blue one, or with the site for the purple one. In this case, let's assume I have only 3. And so this partition sum is just the sum over all possible weight matrices, including the background. The sequence, the sum of all probabilities up to position up to position n minus the length of this weight matrix, times the prior for this weight matrix, so the frequency of sites for this transcription factor, times the probability of the sequence segment under this transcription factor. Right, so this sum you can do for any arbitrary number. And that computational time is going to be linear in the length of your sequence times the number of weight matrices that you have. Also, updating all these priors for all the transcription factors can be done using the exact same expectation maximization technique. So you find if you take the derivative with respect to one of these pi's, then the equation for the optimum is that the frequency pi for this weight matrix is equal to the expected number of sites for this weight matrix divided by the sum of the expected number of sites for all weight matrices, including the background is one matrix. Okay? And you can also use this to tweak your weight matrix to make them better. So instead of taking the derivative of the total likelihood of your data with respect to a pi, you can also take the derivative with respect to one of these components of the weight matrix. And if you do that, you find that at the optimum, the equations that you have to satisfy is that the weight matrix entries, right, so the probability of a letter alpha at position k is equal to the expected number of sites that have letter alpha at position k divided by the total number of expected sites for this weight matrix. All right, so this works in the same way, and you can do this EM, where you start with a random guess at your weight matrix, calculate the total number of expected sites for this weight matrix, and the expected numbers of sites with letter alpha at position k, for each alpha and k. Then you use those to update your weight matrix, and you again iterate. Here I should warn you that here there is no guarantee that you will go to the global optimum. This procedure will only go to a local optimum. And so it's important that you start such a thing from already reasonable guesses at the weight matrix. So people have devised schemes for basically reseeding this algorithm at many, many, many different starting guesses of weight matrices, and then looking at the best ones you found at the end. All right? Sorry? Okay, so what it means is that the probability of the data set, under the assumption that this is a mixture of background letters plus sites for this weight matrix, is maximized.
What will happen if I use an optimal and if I use a suboptimal matrix? Like, how will my outcomes change, so to say? Like, because you started with a
it depends on how bad it is, right? I mean, if you make a weight matrix that says all letters are equally likely at all positions, so then it's going to say, you know, that I can't find any sites, right?
No, you started with the abundance weight matrices, like how you did for the FRUAR.
Yeah.
Yeah, so that was good enough to start with, but then you are using results to now optimize it.
Okay, so I was hoping to not get into this. So, so you're saying, how do you even make these weight matrices to start with?
No.
Because I gave you one case where I knew the binding sites and I made them, and then I went to a case where I have just a large collection of sequences, and I sort of said that you can use that tool to optimize weight matrices, and now you're confused, no?
So basically, I'm trying to understand, now that you have one set of answers and you want to iteratively make your weight matrix better, but are you sure that it is making it better? What is better biologically? Is it so for each organism, maybe the weight matrix may be different?
Let me give you one answer that might satisfy you. So let's say I have a weight matrix that I constructed from a set of known sites, but I only had 20 known sites or something like that. So now somebody did a ChIP-seq experiment where so you know what that is. Okay, so now you've got 2,000 binding peaks in the genome. Each of these binding peaks is a length sequence of maybe length 200. And you know, somewhere in there is at least one binding site. Might be 2 or 3, but let's say at least one. I can now ask, how well does my weight matrix that I have explain that result? Can it explain that when I fished for binding of my transcription factor, I found all those locations in the genome and no other locations in the genome? I can calculate that. And now I can ask, can I maybe tweak my weight matrix to make that probability more likely, to increase that probability, to say, I found a tweak of my weight matrix that now makes it more likely that if I were to randomly sample binding sites, I would get the set of sequences I've actually observed in my ChIP-seq. So that's, for example, what we actually do when we optimize weight matrices.
I understand that, but I would expect that this is an organism-specific thing, and evolution determines that each organism or each species or clades would have a
each weight matrix is for one transcription factor. Each transcription factor has its own weight matrix.
But that would also be different for like the species and clades if you go through. Would it be so conserved that?
Well, okay. So now you're asking, because now you're asking about another transcription factor. You're saying, I have a transcription factor in human, and now I have an orthologous transcription factor in chimp. Do I believe I can use the same weight matrix? If it's chimp, I would say, no problem, go for it. If it's mouse, I would probably still say, no problem, go for it. If it's a fly, and you found orthologous weight matrix, you might want to reconsider. Even though there are certainly transcription factors where you find an orthologous transcription factor in fly and human that have binding patterns that are virtually undistinguishable. Okay? So it happens, but there are other cases where it clearly has changed a bit. So that's, But that's another question, right? They're saying at what sort of time scale in evolution do these motifs change? And there it's sort of, you have to realize if a transcription factor has many targets, then it's very hard for it to change its binding specificity, right? Because if it changes binding specificity, it has to change now the binding sites in many, many, many places, the genome. So transcription factors that target lots of things, they might be kind of frozen. It might be hard for them to change their specificity. All right. So
one quick question here.
Yeah, okay. But I would like to really cover one more thing. So maybe quickly.
So you mentioned that it's we have to be careful that this will discover a local maxima.
Yeah.
And other people have found schemes to start with an optimal start point. So could you tell us a little bit more about that? Are they algorithmic techniques or they're heuristics or?
No, they're all heuristics.
Okay.
So either you may have already some starting guesses for your transcription factor, or you might first start by looking at all words, just actual words in DNA that are overrepresented. So there are quick ways of finding those, and then seeding weight matrices from these words, and then sort of start from there and so on. So there are different heuristic schemes for doing it, but there's no guarantee that you've found the optimum. Okay? All right, so the thing that I wanted to still cover in the last few minutes is what's called phylogenetic footprinting, and that's using comparative genomics to find regulatory sites. So the idea is that related genomes, like a human and a chimp and even a mouse, will share much of their gene regulation. And so many regulatory sites that were present already in the common ancestor of a related species will often have been conserved through evolution. And so one way to try and find regulatory sites is by searching for short sequence segments in intergenic regions that are surprisingly conserved. All right, so you see let's say you have a promoter in a bacterium, and now you find related bacteria and you align the promoters with each other, and all of a sudden you see some segment that is much more conserved than the neighborhood. Maybe that's a binding site. So this idea sort of was around sort of 2000. People started to do these things, and one of the first papers where this was done is this paper from 2003, Kellis et al. Nature paper. So they were the first people to have a set of related yeast genomes. So apart from the genome of Saccharomyces cerevisiae, they had the genomes of Saccharomyces paradoxus, Saccharomyces micete, and Saccharomyces paianus, which are other yeasts. And so here's a picture from this paper where they took the intergenic region between two genes, GAL1 and GAL10, so these are two genes in yeast. And this DNA is the DNA between these two genes. And this is a multiple alignment of the genomic sequence from these 4 species. And so what they did in this paper is they had a simple rule. They just went column by column, and if all the letters were the same, they put a star. All right? So just every time when the letter has not been changed at all between these 4 species, I put a star. And whenever I have enough stars in a row, I forget what the exact rule was, but they had a rule, they say so many stars in so many bases, I predict there is a binding site. All right, so in the red bars here, you see all the places where their rule predicted a binding site. In the red squares, you see all the known binding sites that were known from the literature for this, for these 2 genes. These genes were very well studied in yeast, so many known binding sites were known, and you see that there is a good overlap, right? So the Tata site was found, the GAL4 sites were found, maybe this one was sort of missed, but here was one site missed, and here there was one site predicted where nothing is known, but in general there's a very good match between known binding sites and these places where you find conservation. Okay, so that was sort of the idea that was touted in this paper, but I should tell you that after I saw this paper, I just took another intergenic region at random, and then another one, and played the same game. And I think this one is really not very representative of what happens in most intergenic regions, right? So your mileage may vary with this rule, right? So here's another intergenic region that is well studied. It's upstream of a gene called HIS7. And here are the known binding sites. Two of them, yeah, they are overlapped by a series of stars. But this one is not. And there are lots of series of stars here also in other locations, right, in this sequence. So these approaches are relatively ad hoc, right, with this rule. And so if you have different species that are at different phylogenetic distances and so on, you're going to have to update these rules all the time. So the question is, can you do this a little bit more systematically? And so to do this systematically, what you really need, the key ingredient that you need, is to say, if I have a letter occurring somewhere in a sequence that is part of a binding site, and this letter starts at the ancestor to be at a Y, what is the probability that over a time t in evolution, this letter will evolve into an X? All right, so the basic ingredient you need is along branches of your phylogeny. How likely is it for a specific letter, specific position in a binding site to go from one letter to another letter? All right, so now there is a very fundamental way in which you can approach this problem, and this was something that was developed by Halperin and Bruno, is basically if you know the rates at which mutations are introduced from X to Y, and you assume there is some matrix of probabilities that a mutation from Y to X will be fixed in the population. All right, so the probability that when such a mutation occurs, that it will be fixed in the population. And you now specify that in the limit of large time, there should be a probability Wx to find letter X, right? Because that's what we know. We know that sites that have evolved for a long time have some probability Wx to have letter X. Then you have to pick these substitution rates, these selection probabilities like this. All right, so there is a sort of a general model of setting these probabilities from just knowing the mutation rates and knowing what you want the target frequencies to be in the limit of large time. But actually, in a lot of work, I have used a simpler model, which I think is really sufficient to capture the phylogenetic information. And that model basically assumes that the rate at which you substitute from letter Z to X per unit time, right, depends only on the target letter X. So there is just one mutation rate mu, and the probability to mutate from letter Y to X is just the mutation rate times the final probability, weight matrix probability of letter X. And under that model, the probability that you will evolve from letter Y to letter X over a time t is e to the minus mu t delta xy. This is saying with probability e to the minus mu t, no mutation has yet taken place. And then letters Y and X have to be the same. The ancestor and the descendant have to have the same letter. Plus, with probability 1 minus e to the minus mu t, at least one or more mutations have taken place. And then the probability that I end up with letter X is just the weight matrix entry of X. All right, so this is sort of a minimal model that captures the information. If the letter has not yet been mutated, it has to be the same as the ancestor. If it is mutated, the probability that it's X is just the weight matrix probability. All right, so now if I have an alignment of a certain set of related species, and I see a certain alignment column, I can calculate the probability of this entire alignment column given a certain phylogenetic tree of these species, and assuming that this is a position in a binding site that is characterized by a weight matrix column that gives the probabilities letters A, C, G, and T with this position. And what I have to do is I have to say, well, the ancestor at the root of the tree started with some letter X. I don't know this letter, but it started with some letter X, and that happened with probability Wx. Then along this first branch of T1, there was this probability to go from letter X to Y. Along this branch of length T2, there was a certain probability to go from X to the letter G. This is what I observe. Along branch T3, there was a probability to go from Y to G, and so on. Okay, so for each branch in my tree, I have to put such a transition probability. And now I have to sum over all the unknown letters at the internal nodes of my tree, right? Because I observe the letters here at the leaves, but I don't know what these letters were at these internal nodes, so I have to sum over them. All right, so now again, it seems I have to do a combinatorial sum over these many internal nodes, but Joe Felsenstein has developed a whole bunch of recursion relations that allow you to do such sums on a tree. And so in this particular case, the way it works, how much more do I want to tell you? Well, okay, I'm almost done. 10 minutes, oh, okay. So essentially the quantities that you want to learn, remember that with the previous recursion relations, we said, let's calculate the data up to some position in the sequence. Now what I'm going to say is take a node in the tree and calculate the probability of all the data below this node. And then I'm going to do a recursion relation that basically goes from the bottom of the tree and works its way up. All right? So the way it works is the probability of all the data at some node n of the tree, assuming that that node n has letter alpha, is really the product over all nodes that are children of n. So I'm going to all nodes that are below, directly below n in the tree. Then the transition probability, right? So if I'm here at node n, it has two children. So for each of these children, I have to put the transition probability for the length of this branch to go from alpha to letter beta, and here from alpha to letter gamma with this transition probability, and then multiply it by, if there's a beta here, what's the probability of the entire data under this node? All right, so that's what you see here. This is the transition probability between alpha and beta along this branch. This is the probability of all the data under this node, and then summed over everything that beta could be. Ah, there's a mistake here. There should be a sum over beta here inside the product. Okay? So this is the transition from alpha to beta, the probability of all the data under this child node, assuming letter beta, and now summed over beta, and then the product over all children. So in this way, you can iterate over, do this recursion relation up the tree, and again, in a time that's linear in the number of species that you have, calculate the likelihood of the entire alignment column, assuming that it has been evolving under this weight matrix column. All right, so this is what people developed. So the first paper I know where this approach was used is this paper from the group of Mike Eisen, Alan Moses, where basically when you now look at the binding site configuration that you assign to an alignment, not to a single sequence, but to an entire alignment, then when you score that binding site configuration, you actually, for each column in the alignment, you use this probability of this alignment column given the tree and the weight matrix, and similarly for background columns, which again you should think of as a weight matrix of length 1. All right? So they had a model that slides this over the sequence and gives a probability at each position. And then what we've done is we've basically combined the two kinds of recursion relations and basically allowed to do any configuration of sums of binding sites for any number of transcription factors using this recursion relation to calculate the evolutionary scores at each column and using the other recursion relation to do the sum over all possible binding site configurations. Okay? And you can do all kinds of extra bells and whistles on this thing, but I will not talk about this. So what I would like to just finish with is to say is that right now, with lots of study over the last decades, we now have really a fairly large number of weight matrices available in various databases. So for example, for E. coli, there's a database called RegulonDB that has weight matrices for about 125 or so transcription factors of about 250 transcription factors that E. coli has. The database Jasper, it covers several organisms, but for example has about 500 weight matrices for vertebrates of the about, maybe 2,000 or so transcription factors that exist. In Fly, there are also collections. There are weight matrices for about 140 out of the 600 transcription factors that are available. We also have a collection. I think our mammalian collection is particularly good. So we have about 680 mammalian weight matrices for about 200 transcription factors. And yeast is a particularly nice case. In yeast, is the only species that I know where the weight matrices are available for the large majority of transcription factors. So yeast has maybe 180 to 200 transcription factors, and we have weight matrices for 200. And what you can do is you can use such weight matrices in combination with the methods that I just shown you today to make annotations genome-wide of binding sites in intergenic regions in these species. So for example, so here we have a database and there's a web browser, so you can go somewhere in the yeast genome and you see in this G-standard intergenic region, these are all the binding sites predicted, and it's written what transcription factor is supposed to bind there, and the intensity of the site is the posterior probability that's assigned to it. All right, now I'll tell you that, okay, so I'll skip this, I'll tell you that those computational predictions can actually be shown to be more accurate than the results of, for example, a chip-chip or a chip-seq experiment. Okay, so I'll skip that right now, but you can do direct comparisons of what predicts known sites in the literature better, a chip-seq experiment or a computational prediction. And the computational predictions will in most cases clearly outperform such genome-wide binding assays. I just want to mention this because in many, many cases, biologists insist that predictions need to be validated by such biological measurements and kind of ignore the fact that those biological measurements are very far from perfect themselves. All right? Okay, so the other topic that I was planning to talk about today but don't is, can we now go from these constellations of regulatory sites and understand how they lead to stable gene expression states? Okay? How does the cell read out this genetic regulatory code to determine the gene expression states? And I will just leave you today with my feeling about this after working on this problem for quite some time. So in the psychology literature, there is something called the Dunning-Kruger effect, which basically shows how the confidence in somebody's ability goes as a function of how much they've studied a certain topic. Okay, so along the x-axis is how much you studied it. So here you have not studied at all. Here you've studied it all your life. And along this axis, how confident are you about your abilities to say something? And so here is very low confidence and very, very high. So here are sort of two examples of extrema of this space. And what people have found is that, okay, so if you have never looked at a problem at all, then you're not very confident. And if you start studying it, then you quickly start becoming much more confident about what you know about this topic. But you're going to reach some maximum at some point. And then there's going to be a precipitous drop where your confidence starts decreasing when you get sort of an inkling of the enormity of the stuff you don't yet know. And this is where I am now with respect to how to go from these constellations of regulatory sites to really understand why are there discrete cell types, for example. Okay. Anyway, so that's what I'll leave you with today, and tomorrow I'll switch to another topic.
Okay, so we have time for 1 or 2 more questions.
Yes.
So, Eric, I completely agree with you about the problems with getting binding site information from ChIP-seq or ChIP-chip, but of course you need to have experimental information from somewhere.
Yeah.
And I was wondering what you like, footprinting, CellX, Matomi?
Well, I think that there are different benefits for these different. So I like ChIP-seq because it tells you something about where that thing was. It might have not been binding the DNA directly. It might have been binding the DNA and not really doing anything. But at least you know that in vivo, in some cell type, this thing was there at some frequency. So that's, I think, the benefit of ChIP-seq. I think the CellX data has been very good in sort of increasing the number of transcription factors for where we have at least some rough model. Okay, so it's been more successful than anything else for that. Footprinting is great, but typically you don't get that in high throughput. So there is just it's a problem of scale. It's hard to scale it up. So I mean, we're just using whatever combination we can get our hands on. I don't think it's an insolvable problem, right? So if you look at yeast, I think for most transcription factors there, these models are really not so bad. I mean, we recently did a study where we did much more fancy models that have correlations between positions and all these things. And for 80% of transcription factors, you don't need it. Okay, you really cannot outperform a simple weight matrix. So I think in most cases, this is quite good. But there are cases like, I know when I moved to Basel, Walter Gehring asked me to use his footprint data together with some other assays to make a weight matrix for ILS, which is the master regulator of eye development in fly. And this was one of the biggest failures of my life. Okay, could not do it. So there are transcription factors that have some complicated different modes of binding when they have different partners, different cofactors. And so there are cases that are much more complicated. And I don't even know how to really address those because some CellX thing where you just express one binding domain is not going to catch it for you. So, so I don't know. That's basically the...
Yeah, I had a quick question about the using evolutionary trees to refine WMs and also derive them. Are you actually able to track the evolution of new binding sites and lineages? I mean, that wasn't clear from... Yeah,
so you can always make things more complicated. So when we've done this model, right? So we've done a model and says either this site was evolving under the weight matrix along the entire tree, or it was evolving according to the background model along the entire tree. But of course, you can make additional models that say, well, this site started in this clade, and it was evolving under the background model in all the branches, except for the ones in this clade. Okay, so we've done such things where you look at what are places where you can start or remove a site, and does it increase your likelihood, and you can try to even sum over that if there are not too many possibilities. But of course, at some point, there's always a trade-off between the richness of your model and the extent of your data. And at some point, if you have enough data and you're going to include hypotheses that the sites may have been gained and lost many, many times, at some point you can't infer anything anymore. And probability theory will tell you, well, you've given me so many options, I can't decide, right? So that's sort of the trade-off.
Might be an unnecessary complication, as you have said, but amino acids, I mean, let's say you're working with, is there a way to easily incorporate that information?
Okay, so there are people that have tried to say, well, can't I just look at the DNA binding domain, look at the protein, and somehow predict from the protein what is the sequence that it likes to bind? So that turns out to be, let's say, not so easy. So so I know there are some attempts to do this for zinc fingers where people have reported that they had some success in this. I've not been completely convinced that it's really been that successful. There have been other cases where if you have a co-crystal of the transcription factor binding the DNA, people have played with then changing the nucleotides in the co-crystal and making predictions how that would change the binding affinity. So that certainly does much better than random. And that can be used to give at least some sort of guess at the best binding sequence. All right? Not the whole weight matrix, but the best binding sequence. The sort of downside of this is, it's good. The downside is by the time that this thing has been studied to the extent that you have a co-crystal of the transcription factor binding inside, you typically have a collection of known sites already. And you would have known the consensus from that also. So in the end, it doesn't really add a lot of information. And I'm not aware, there might be something new that I'm not aware of. I'm not aware of somebody really being able to accurately predict binding specificity directly from the protein sequence. I would love to do it, but yeah. 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 Right, so this has been, so I would say this has now been extensively studied, and it's certainly not true, okay? But it's a very good approximation for most transcription factors, and also the approximation tends to be good for sites with high energy. Like so, let's assume I started the site with the best energy possible. And now I do 1, 2, 3 mutations and I lower the energy. The linear model is very good in predicting how much your energy will go down, okay? By the time you go all the way to a random sequence and you've changed everything to be bad, now your model will predict that there's almost like a repulsion and you'll say, oh, this transcription factor will really hate this sequence. Well, in reality, it doesn't really hate this sequence. So the linearity sort of breaks down in the low binding energy regime, not in the high binding energy regime. And given that we care about the high binding energy regime for finding these sites, you get away with it. So this sort of, I think most of the time you can get away with it.
Which binding site have different energy levels?
I think that's just a fact. Different sequences have different binding affinities to the transcription factor.
So does it mean that the different binding sites do not interact with each other?
Sorry, can you repeat that?
Please, does it mean that the different binding sites do not interact each other?
Well, they may, right? I mean, it depends on what you mean interact, right? A binding site is just a piece of DNA. So if there's a piece of DNA here and another piece of DNA there, then
what I want to say by interaction, I mean that the different reactions happening in a specific site does not affect the activity by the other side?
Yeah, it does. It can. So it can in various ways. So one site may bind. This may now stop other sites from binding their overlapping sites. That's a trivial one. It may also be, there's another site over here, and it turns out that they can touch each other, those transcription factors, and they like each other, they have binding affinity to each other, and they actually help each other bind to the DNA. Or they might not like each other, and they may inhibit each other from binding to the DNA. This happens when they're close enough to sort of physically touch these transcription factors. There is a third thing which I will mention tomorrow, which I think is very important, is that in eukaryotes, all the DNA is covered by these nucleosomes, right? These nucleosomes, they cover about 150 base pairs of DNA each. And now it happens that transcription factors have difficulty getting to their binding sites because these big nucleosomes are in the way. And now if binding sites occur under the same nucleosome, they can help each other, right? Because if different transcription factors are trying to bind at the same time to different sites that sit under the same nucleosome, they may effectively, people call this passive cooperativity, help each other's binding. So that's sort of like an interaction that I think is actually really important in eukaryotic gene regulation. But those are the main things I can think of in terms of interaction.
Okay, it's good.
Right?